with you virtually. I think we now are on 3 continents together, you in Europe and me in North America and the audience in South America. So thank you very much. I'm going to start to share my screen and we will do that, and I'll get my presentation loading, and I will just ask someone to confirm that my screen can be seen, and then I will start.
Yes, we can.
Okay, so thank you very much. It's a pleasure to be with you today. I'm going to share some of our work on engineering T cells and redirecting the specificity to cancer. First, I have to declare a financial interest due to intellectual property and patents in the field of cell and gene therapy, and those are listed here, and the conflict of interest is managed in accordance with University of Pennsylvania policy and oversight. So the problem that is that there are challenges in cancer immunotherapy where the immune system evolved to tell self from non-self. And since cancer is derived from our own cells, it's not only looking like looking for a needle in a haystack. Because of the evolutionary mechanisms that cancer uses, what we find often is a dull needle. So how to find that needle and ensure it is sharp is the mission. And so we can approach that a couple of ways, and we can look at the endogenous T cell receptor. Here on the left, it's a heterodimer that recognizes antigen presented by self-molecule MHC. We can search for a T cell receptor that recognizes a cancer antigen, those that have been identified. But this transgenic T cell receptor, while it recognizes intracellular antigens, it is MHC restricted. So you must identify this construct for each tissue type. The chimeric antigen receptor is a hybrid antibody binding antibody domains on the outside that recognize surface antigen and T cell signaling domains on the inside. This is MHC independent, such that the construct is off the shelf. Now, only about 1/3 of cancer antigens are presented on the surface, so T cell receptors still have some advantages. Now, if we go back to the very beginning of chimeric antigen receptors, they came about in the late 1980s when the structure of the B cell antigen receptor, or the antibody, was being elucidated along with the structure of the T cell receptor. And 2 groups, one in Japan and one in Israel, said, well, what if we mix and match the parts? What can we learn about T cell biology? And here are some figures from those papers, the constructs on the left and some functional data on the right. But what really struck me in reading these papers is in the discussion, where they say in the future it might become possible for T cells recognizing any antigens without MHC restriction to be produced by the technique described here. And this approach can be exploited to direct cytotoxic T lymphocytes to kill tumor or virally infected cells. So I find it really prescient, amazing that each of those groups was thinking decades ahead of time. And where are we now? Well, we have 6 approved CAR T cell products in various jurisdictions. We also have, in the past 2 months, 2 FDA-approved gene-modified stem cell therapies, one for beta thalassemia and one for adrenoleukodystrophy. So the emergence of these types of genetically engineered therapies to treat disease, to redirect immune specificity, will be proliferating in the near future. And it's possible in the US we could have another approved product, a virus-specific product, but in 2023, I believe there will be 3, tabelecleucel, if not in 2022, and then afami-cel and lifileucel, which is a tumor-infiltrating lymphocyte product. Now, earlier this year, we presented decade-long follow-up on our leukemia CAR T-cell trials, but we actually started in HIV. That's shown in the top left, where we showed decade-long persistence of CAR T-cells in trials in HIV subjects. Now, earlier this year in leukemia, and let me tell you a little bit about the findings there. You see here on the top left flow cytometry data showing persistence of these CAR T cells. And the two graphs below that show the qPCR data along with the flow cytometry data in the red triangles and the blue circles, and the length of the B cell aplasia is shown in the blue squares. There are other ways of tracking these cells. If we look in the middle, the blue circles, this is looking at the number of copies of micrograms of DNA in molecular tracking. Also integration sites, and that's shown in the next graph. And these two patients on which we have the longest data show slightly different patterns in their integration sites. So there's not a particular integration site that we can correlate with this long-term persistence. We also went back to the freezers and performed more advanced phenotyping than we had originally, using a 40 antibody panel that you see here. And by cluster analysis and longitudinal analysis of samples, we could show that in the beginning, immediately after infusion in the earlier term, in the months and a year or two, we do see an initial response composed of CD8 positive cells and some cells that are gamma delta CAR T cells that go away. But in the long term, we see a predominance of highly activated CD4 positive CAR T cells, which was a surprise. And these CD4 CAR T cells displayed evidence of ongoing proliferation at the transcriptomic level and also by Ki67 staining. They upregulated activation markers, CD38 and HLA-DR. Also, inhibitory receptors were upregulated. And by gene set enrichment analysis, we saw significantly upregulated genes in CAR-positive cells compared to CAR-negative cells that you see here. And reanalysis of previously published flow cytometry data showed the functional capability of these CAR-positive cells, and also that CD4 negative, CD8 negative population that was gamma delta T cells. So what's done here that you can see on the right is the samples from the patients are added to CD19 expressing target cells, and you see the specific elution of CD107A, which is a marker of degranulation, and the chemokine MIP1 beta. So to summarize the paper here, which has much more data than I have time to present to you, there is an initial period of proliferation where we do see CD8 cells predominating, but in the long term, we see the CD4 cells and continued proliferation accompanied by increased oxidative phosphorylation, mitochondrial biosynthesis, and aerobic glycolysis. So that's another thing that we've observed here and previously, is the metabolic changes that accompany the immediate near-term post-infusion period and the long-term period. So there are these two phases of engraftment and persistence. I mentioned the gamma delta cells and the CD4 positive population. We don't fully understand the reasons for this long-term oligoclonal selection. It may or may not be disruption of nearby genes or integration into genomic regions associated with robust CAR T cell expression, but we still don't understand why or the possible role of genetic drift. Now, I want to talk about the raw material, and I think I mentioned this last time I spoke to this group. We have to recognize that not all patients, not all donors, if you're thinking about healthy donors, not all T cells are created equal. This is a model that has been popularized by Luca Gattinoni, and shows the differentiation from naive T cells to stem cell memory, central memory, effector memory, and short-lived T effector. And while you do have enhanced effector function towards the right-hand side, you also have senescence. And so it's generally agreed in the field that the types of cells that we're looking for in a product and to generate the greatest potency that is durable is a stem cell memory and a central memory phenotype. And the question is how to get there and what to do if you don't have those cells at the beginning. Because we're presented with this conundrum. The approvals so far are in relapse refractory patients, and we are getting patients that have undergone many treatment cycles. And what you can see here is the greater number of treatment cycles, the higher the predominance of this gray subset, which is the terminal effector T cells, which are the ones that you don't want. And not all collections and manufacturing runs are predictable. I can share with you that perfect can be the enemy of good enough. And the question is, what is good enough? So let's talk about raw material. How do you know that you always have the right cells and what cellular material is acceptable? I think this applies to many different types of cell therapy. Look at the bottom left at that color palette, and I'll ask you 3 questions. Are all of those colors blue? Are all of those colors blue enough? And do all of those colors have blue in them? So how do you think about qualifying the cellular raw material? Well, we can think about improving T cell quality in several categories by different methods of activation, by adding cytokines to the culture, perhaps in vivo, by inducing metabolism and transcriptional regulation and gene knockouts. So we want to teach every T cell to be all that it can be. The data on improving cell function is coming in fast and furiously. I first made this slide about a year ago, and I started gathering reports on data that was coming out on ways to enhance potency, and pretty soon there were more reports, and then last year at ASH, even more. And I have this list now that is about 25 methods that you can either knock out a gene, add something in, or make changes to the culture. And now the question to consider with all of these reports, and most of them are preclinical, some are in the clinic, but with all of these reports, how do you know which is best? What if there's a combination that is best? 2 to the 25th is a large number. So I think it's a good thing that there are many clinical trials going on around the world on engineered immune cells so that we can better understand how we teach every T cell or every immune effector cell to be the best that it can be. And so we are in the era of synthetic biology. With respect to CARs, we can create logic-gated Boolean CARs that function in an and or not fashion, checkpoint-resistant CARs, safety switches, and conditional and stealth CARs. And we're going to need all of those to address the challenges of solid tumors. And I'm going to present one of those methods here, and it's a so-called armored CAR. Now, prostate cancer evades the immune system by prostate cancer cells secreting high levels of TGF-beta. What does TGF-beta do in the tumor microenvironment? Well, it suppresses T cells. Now, one strategy to get around that is to include in the CAR construct a dominant negative TGF-beta receptor that serves as a decoy, so it's unable to signal, but it can soak up TGF-beta in that microenvironment. And so we've been testing this in a clinical trial in prostate cancer targeting PSMA, sponsored by Community. And this was recently published, and the results I'll summarize for you here. On the top left, we're looking at the engraftment, and this is a multi-cohort study, a dose escalation, the inclusion of conditioning chemotherapy, and then the inclusion of conditioning therapy with a dose de-escalation due to limiting toxicity. In the top right are the levels of TGF-beta. You can see some are steady, some go down a little bit. And I want to present one patient in particular, and you can see the pretreatment disease burden here on the left. Now, in the middle top, we're looking at the presence of the CART PSMA cells, and you can see that dramatic increase that is accompanied by a decrease in serum PSA or prostate-specific antigen, and also a decrease in the serum TGF-beta. While that expansion was going on, we see an oligoclonal emergence of T cells as measured by the T cell receptor V beta families, and also by looking at the lentivirus integration sites. So this is our way of fate mapping the T cells by these two methods. The clinical data is shown here. The graph on the left is the PSA level, how that decreases, and you can see patient 9 here had this very dramatic decrease in PSA as well as the TGF-beta, and we had a number of other patients that are showing this decrease in PSA, and on the right is a scan to show the disappearance of a lesion here at month 3. Now, that one patient did have toxicity, cytokine release syndrome, that was resolving, but unfortunately the patient developed sepsis and passed away from that. But we do have evidence that this functionally armored CAR is feasible, generally safe, that we did do a dose adjustment following that toxicity. There is a dose-dependent and lymphodepletion-dependent relationship between expansion and inflammatory cytokine expansion and cytokine release syndrome. And the peak levels in the blood are comparable to what we see with CAR T cells targeting CD19. So we have early evidence for anti-tumor activity. We have a PSA 30 decline observed in about a third of the patients. Next, I'd like to present to you another strategy using a synthetic receptor that signals in a way that can be controlled. And this is using synthetic IL-9 receptors. This is work from Carl June and Chris Garcia and Miko Sierrala. And the construct here is a little bit different. It's using on the extracellular domain a modified IL-2 receptor beta, a so-called orthogonal IL-2 receptor beta that is mutated and cannot bind wild-type IL-2, but only binds ortho-IL-2. And the design of the experiment was to see if this construct signaled, that's the base case, and then compare it using different cytokine signaling domains, namely IL-7 receptor and IL-9 receptor. And since IL-9 is not normally found in T cells, this was the real question mark. What would the phenotype of this be? And so interestingly, what you can see here is when we compare in this pancreatic ductal adenoma carcinoma model, this is the IL-2 receptor, the orthogonal in green, and this is the IL-9 receptor, and you can see that it's significantly better. So the design here would work this way. We deliver this orthogonal receptor to T cells, and then you can deliver the orthogonal IL-2 to activate the receptor by an adenovirus to solid tumor. And that I should back up a little bit and say, why are we using orthogonal IL-2? Because with natural IL-2, you activate regulatory T cells that suppress the immune system. And so the bottom line here is that we find that the IL-9 signaling domains augment effector and stemness of CAR T cells and T cell receptor T cells. Now, what about the potency of off-the-shelf allogeneic products? Can that be improved? We have to qualify those as well. I'll present here one underappreciated and underexamined aspect of autologous CAR T cells, and that is, unlike the allogeneic CAR T cells, the autologous retain their endogenous T cell receptor. And what that means is there is the potential for epitope spreading once you have lysis of tumor. And that's demonstrated here in this mesothelin-specific CAR T cell trial, where we see the induction of anti-tumor activity. So I want to move on now to talking about how we're manufacturing improvements and faster CARs. And this is work that originated from Saba Ghasemi and Mike Malone, where to our conventional 9-day process, they compared a 5-day process and a 3-day process. And on a per cell basis, you can see that the day 5 are more potent than the day 9, and the day 3 are more potent than the day 5. Based on that, we've opened up a clinical trial in patients with CD19 hematologic malignancies with a twist that also includes an IL-18 secreting construct. This is a report from a Chinese company, Gracell, a couple of years ago on a 1-day process. The stars here are marked complete responses. And they had an update at the EHA meeting this spring in 29 patients, 27 demonstrate a response. That's significant. I think this needs to be confirmed, but there are some other issues when we think about a 1-day process that I'll get to in a moment. And this is data from ASH 2021 from Novartis showing clinical data in a 2-day process in a clinical trial in multiple myeloma and also in diffuse large B-cell lymphoma. And the clinical responses are here in a small number of patients on the right, this in vivo cellular kinetics on the left. And what is quite interesting here is that there is comparable in vivo expansion at a 25-fold lower dose compared to the regulatory approved product, Kymriah. We published also Saba and Mike Malone on a 24-hour process. So I think you're going to see some of these new manufacturing processes for CAR T cells and clinical data and a way to address some of the bottlenecks, perhaps. Well, what are the issues here? I show our quality control release testing for our conventional CAR T cell product, where we're testing viability and phenotype, residuals, endotoxin mycoplasma, transduction, and so on. Well, for a 24-hour non-activated product, how do you think about CAR expression where the gene may not be fully integrated? How do you think about vector copy number? And how do you think about the assay for replication competent lentivirus? And perhaps most challenging, how do you think about a potency assay and what will the regulatory agencies accept? I think all of those are unknowns at this point. And so CARs are moving beyond T cells. We have clinical trials and reports of CAR NK cells, CAR macrophages. People have put CARs in blood stem cells and CARs derived from iPSC. And T cells are more than CARs, of course. We have these reports of T cell receptors and TILs. And CARs are moving beyond oncology. We have CAR T cells actually started in HIV. A very exciting report in September from a German group, CAR T cells targeting CD19 in lupus, and some preclinical studies, CAR T cells targeting cardiac fibrosis and potentially for aging. Now, we are doing autologous. You've heard about allogeneic, and I've mentioned iPSC. And the argument is made that this can provide CAR T cells for the masses. But what if we are able to leapfrog allogeneic and iPSC by an off-the-shelf autologous approach? Well, what is that? That's shown here. And there are billions of doses of lipid nanoparticle encoded messenger RNA that have been given to humans. What if we use lipid nanoparticle-coated messenger RNA to encode a chimeric antigen receptor? And what if we direct that lipid nanoparticle with an antigen directed against T cells. And that is this report from the University of Pennsylvania from January, targeting fibroblast activation protein in cardiac fibrosis. And this was demonstrated in vivo in mice by luciferase expression, specific expression in the spleen, but not the liver, and also functional expression by delivering a Cre recombinase cargo. That data is shown here on the left. And really striking that you can get 20% expression by the delivery of these CARs in vivo with no ex vivo manufacturing. Here's a model of cardiac fibrosis. You induce fibrosis by the infusion of angiotensin II and phenylephrine, and the repair following delivery of the CD5 targeted lipid nanoparticle FAP CARs is shown. The baseline control is shown in gray, that's saline. The induction of fibrosis is shown in the tan, and following delivery of the CARs is shown in the red. And here's another figure showing that same principle. And so we look for the further developments of engineered cell therapies, but what are some critical path issues for wider access? We need to work on enhancing potency, especially for solid tumors, by incorporating some of these tools. Ex vivo manufacturing is complex. We need to shorten the culture and integrate automation. We need education and training at all levels. Patricia talked to you about IACT. I'll remind you that IACT is built on 3 pillars: a scientific pillar, a regulatory and quality operations pillar, and a commercialization pillar. And much like the Rosetta Stone, to be able to translate, you need to be proficient. You need to know the language of all of these. And please join us in Paris, May 31st to June 3rd, 2023. Finally, we need to recognize that these therapies are financially complex and work on affordability by integrating predictive biomarkers and value-based payment that links price to outcome. I will close by showing you our first pediatric CAR-T patient, Emily Whitehead, in mid-April 2012. 10 years later, May 2022, she is cancer-free. She's an honor student. She has her driver's license, and she is currently in her senior year of high school, applying to colleges. Emily's story is unique and exceptional, and there are many twists and turns to it that have been captured in a full-length documentary film called Of Medicine and Miracles. It's playing at film festivals in the US. Unfortunately, not that I'm aware of abroad yet, but hopefully if you're in the US, you'll have a chance to see it, pending distribution internationally. And I am a representative of a very large team at Penn Medicine Center for Cellular Immunotherapies, recognizing our collaborators, our funders, philanthropy, industry, and especially our patients. It takes a village, a huge team, to bring these therapies to fruition. So thank you for your attention, and I will stop sharing.
Thank you, Bruce, for your fantastic talk. Every time that I listen to you, I am very, very happy. So since this is an opening ceremony and we have your full conference, there is no questions, even though I know that the ABT cell members, everybody in the meeting, all of them are very excited to do several questions, but they can send email, no problem. And so I'd like to thank you so much. Now I would like to invite all of us, all of you, to the opening cocktail in the exhibition area. Thank you so much for being here with us, Bruce, and thank you so much, Dr. Sang, for the invitation, and we are very, very happy. So, bye-bye for everybody, Sang.
Thank you.
Thank you, Bruce.
Thank you, Patricia. I wish you safe travels home. And
Yes, sure. I will be back to Brazil.
yes.
Thank you. Professor Sang, do you want to say some words?
Now, I just want to thank very much Dr. Bruce for accepting our invitation. It was really nice talk. And I saw several times your talk. And as Patricia said, every time I watch your talk, it motivates me more to work in gene therapy. Thank you very much.
Yeah.
So
Thank you,
you. Bye,
Sang.
Bruce. Have a
Yeah,
safe
thank
trip
you.
back home.
Thank you.
Thank
very
you.
much.
Bye,
Bye-bye.
Sang.
Bye.
Thank you so much for everything.